Okay, so hi, my name is Eric. I'm happy today to present my project, automating linear motif predictions to map human cell signaling networks. So I'm Eric. I'm from McGill University from Canada. I'm an undergraduate student in bioengineering doing my last year. So today I'm going to start my presentation. So what are the short linear motifs? The motifs, they are short protein sequences, short protein stretches composed of less than 10 amino acids in the proteins. They usually mediate transient interactions, transient protein-protein interactions as opposite to permanent ones. Somehow it's due to their characteristic of being in the disorder region in the protein. So disorder region means those regions that don't have a fixed structure. The structure can change from time to time, and somehow this is kind of reason why they mediate transient interaction. Because they are in disorder region, it's also hard to determine them experimentally. So methods like cryo-EM or affinity capture, they don't work with transient interactions optimally. The base model that we're using now in this project is called Gibbs sampling. This is not something new. Previously, even in 1993, people have already started to use Gibbs sampling in linear motif discoveries. And then 20 years later, two of the most, the best people in this field are MIM-Suite and SlimFinder. They still use this base model to discover new linear motifs. In this animation on the left, you can see how it works in protein searching. Basically, you sample random windows. You accept a new window if it is better, and you reject window if it is worse in order to converge to the final global minimum. Yeah, this is a global minimum algorithm instead of local minimum. So usually the Gibbs sampling, they use a matrix to represent a motif. It's called PSSM, the position specific scoring matrix. It's a very smart way to compress all the data of a motif into numbers. It's also the reason why they use Gibbs sampling, because Gibbs sampling require many samples throughout the process. So we thought about, can we insert some gaps in the PSSM? It turns out that the hidden Markov model can do the thing here. You can see on the right that the hidden Markov model is able to capture insertions and deletions instead of the fixed rigid window size. Now the question becomes, if we use hidden Markov model in there, then we have to train a hidden Markov model every time we sample it. And then we have to sample it throughout the Gibbs sampling thousands of times. So that means thousands of hidden Markov model need to be done during the search. And that's tremendous computational time. So how are we going to deal with this tremendous computational requirement? We use bias sampling. Basically, we look at the protein regions that are more likely to contain motifs. And how we did it, we look at 4 feature scores. The first one is using the language model, protein language model, ProT5, by Professor Rost in 2021. It was a very big improvement in this field of the ProT5 language model. We also look at the conservation score and the other 2 structure information, disorder and the solvent accessibility from AlphaFold. So this is an overall of our tool. I skipped the data curation because of the time limits, but we use all the tool to validate with the ERM database, which is an experimentally curated database. Some of the results, we can see that very surprisingly, if we look at the contingency table on the right, we capture almost half of the experimentally validated motifs in the ERM database. So that's a big improvement, actually. Almost half of the experimental data are captured using our algorithm. Some case studies, this is RON-X1 is a protein, is a transcription factor used in the blood cell production. We find a motif on the end of the RON-X1 protein with its partner kinase, MAPK1. And then and all the scores are above at least 1 standard deviation, Gibbs sampling works very well to find this new motif. This is also a protein known for a process in the leukemia. So we have this discovery algorithm done, and we are now building a database to allow biologists to use our data set. So thank you very much. That's all for the presentation. I'm happy to take questions in my poster session.
Thank you very much.